Kijas from CSRO, who is going to join us soon via Zoom, and he's going to be chatting about genome-wide chromatin accessibility and transcriptome profiling during onset of maturation in Atlantic salmon. So let's hear James.
Hi everyone, my name's James Kijas and I'm from CSIRO here in Australia. I'm going to talk about our study in multi-omic profiling of Atlantic salmon today. The program said that my colleague, my postdoc in the lab, Ayman Mohammed, who you can see here, was going to be at the talk, but unfortunately he's not able to, so I'm standing in and I'll try and do as good a job as Ayman would have. Well, I'm introducing people. This is Marina Navarre-Sanchez, Antonio Roberta, who contributed to the study. This is my late colleague, Harry King, who really conceptualised and started the work off, and our industry colleague, Brad Evans. Without him, none of this would have been possible. So the topic that I'm going to talk about today is sexual maturation in Atlantic salmon. So this is a fundamental life history trait. It's ubiquitous to all vertebrates, so it's of wide interest. But it's also critically important for efficient Atlantic salmon farming, at least it is in Tasmania and Australia where we are. The reason for that you can see here. So if you look at harvest age animals, 22 months of age, fully half of males will have already undergone the maturation process, and this renders them unusable as production animals. So it's a really important trait that we want to understand at a molecular fundamental level. It's also interesting in so much as if you think about some of the organs in the body, it undergoes this really quite radical physiological reshaping. So we reasoned it also might be an interesting model to explore epigenetic control, and that's largely what I'm going to talk about today. So our aim in this work was to identify the earliest biological triggers of sexual maturation. Pretty easy to get unmaturated or immature animals, compare them with ones that have already matured. But we didn't want to do that. We wanted to target the earliest commitment signals that send animals on this trajectory. So we conducted an animal trial, which I'll tell you about briefly on the next slide. We collected 3 types of data, which I'll tell you about, and then we intersected them to see what we could find about the control pathways for early commitment into maturation. Okay, so this summarises the experiment that we did. So if you think about the signals that commit animals to enter sexual maturation, for Atlantic salmon, it's increasing day length, the day length on the Y, and animals will commit as they approach the summer solstice. So what we did was we took 3-year-old female animals, and at the winter solstice, we put them straight onto a long light regime, such that in the experiment, time point 1 was before the initiation, immediately before the initiation of this long light, and then we took 3 time points, T2, 3 and 4 following. And so the structure of much of the analysis is comparing these T2, 3 and 4 time points to this baseline before the long light went on. So we had an early indicator that the animal husbandry here in the experiment was a success. Using gametosomatic index. So from females, this is simply the ratio of the weight of their ovaries to their total body weight. And you can see at T3 and T4, that's going up. So their ovaries are getting bigger. Okay, so this is the data generation. There's a lot on this slide. I mean, the take homes are time points 1, 2, 3, and 4, and 3 data types. So RNA-seq. We also collected whole genome bisulfite sequencing to assess genome-wide methylation status. And lastly, we conducted ATAC-seq to look at chromatin accessibility. And so the bars here show which samples have all 3 data types, which samples only have 2 or 1 data type. And I'll come back to that as we go through. So, I mean, that's a multi-dimensional, pretty complex data set. So what I'm going to drill into today really is an assessment of the dynamics between the data types. So here are the 3 data types, transcriptome, methylome, and chromatin accessibility, and show you some results about the strength of epigenetic control of these 2 types of data on the transcriptome. again, in the context of this time course, which has a biological event occur. So I'm going to provide you some global trends. I'm going to sort of steer away from a whole lot of down in the weeds explanation of specific genes. We've got a lot of them, but I'm going to focus on some global trends and three things really in particular. First, we'll just look at the transcriptomic remodeling that occurred via maturation as a single data type. Then the relationship of methylation changes to those gene expression changes. And then lastly, some comments on the chromatin accessibility from ataxia and how that related to gene expression changes. All right, so first up, I'll explain the transcriptomic component. And so on the next slide, you're going to see 9 panels. So 3 tissues: pituitary, ovary, liver. Brain isn't there. I could go into why, but basically we weren't happy with the amount of variability between replicates compared to the amount of variation in the data set. It was too high between replicates, and so we discounted it. So there's 3 tissues: pituitary, ovary, and liver. T1 versus T2, T1 versus T3, and versus T4. So time goes in that dimension, and then tissues in this dimension. And so this shows really, I guess, a summary of the transcriptomic outcomes. So if we think of pituitary, or look at pituitary in the top 3 panels, early, time point 1 versus 2, very few genes. And in fact, in the pituitary, we didn't really see much up and down regulation. So a small number, a couple of hundred upregulated genes and just 70 odd downregulated genes. And that's quite different to what we saw in the ovary. So here, this tissue has undergone a fundamental remodeling across the time course. The tissue has just basically exploded into transcription, such that by time point 4 compared to time point 1, there's 3,500 upregulated genes and a couple of hundred downregulated genes. The third tissue is liver. And here you see this balance of up and down regulation, and you don't see this increase in differential behaviour through the time course, which is perhaps to be expected for a tissue like liver for a trait like sexual maturation. So as you can imagine, we delved into, did a regular gene ontology enrichment type analyses of these data, and we found a bunch of stuff that you would expect to see. So in the pituitary, primarily hormone receptors, growth hormone and its subpart receptors, steroidogenesis genes, sex-related transcription factors, and so forth. And I could bore people with this for hours. There's a lot of biology in here that you would have expected to see, some of which we didn't, in fact, expect to see. So we saw quite strong sets of genes involved in the inflammatory response in the liver, in the changing ovary. And in fact, with some reading, that's been observed before, that there is an inflammatory response as the ovary goes through these remarkable changes. All right, so for the rest of the talk, then, I'm just going to talk to you about, well, what relationship did other epigenetic signatures have with that gene expression profile change that I just showed. And we'll start with methylation. So just to refresh your memory from earlier on, we've got methylation data at T1 and T4, so just the beginning and the end of the experiment, not in between, and we've got it for all 3 tissues. So we call differentially methylated regions, or DMRs, and you can see the threshold that we apply here. The first one is that there actually weren't that many of these DMRs genome-wide across this time course. So there were nearly 3,000 in the ovary, and the ovary had the most of these, which perhaps is interesting because it's the effector tissue for the trait. The other observation is that they were tissue specific. We saw very small overlaps in these DMRs between tissues. So the methylomic changes that we see appears to be tissue specific. When we look at the genomic position of these DMRs, they were strongly either in genes or introns or within 5 kb up or down stream of genes. So there were only 30% of them that sat out or were intergenic between genes. So that gave us really strong early confidence that the data set's got biological sense. It makes sense because we know methylation should be occurring next to genes. Now, of course, because it's differential, it has directionality. So at T4, samples could either be accumulating higher levels of methylation and be high per-methylated, or the opposite in T4, where they're losing methylation and be hypomethylated. And we saw that the balance in these DMRs between hypo and hyper was about the same for the pituitary and for the liver. But we saw this quite strong bias towards hypermethylation in the liver across the different gene ontology, gene annotation classes. So it appears that in the ovary, as maturation occurs, it's accumulating higher levels of methylation. Okay, so to the meaty bit, which is, well, what's the relationship with gene expression? So this shows all of the genes in the data set at D2, where you have differential gene expression on the y-axis and the average expression on the x-axis. And then we overlaid onto that differential methylation for those genes where it occurred in the gene. And we saw no relationship. There just wasn't any. So an early conclusion here is that global patterns of methylation appeared in this data set not to be strongly controlling gene expression. So next we said, well, all right, so let's just have a look at those differentially methylated genes and assess their transcriptomic behaviour. And similarly, there was no relationship. So even for DMGs, we didn't see an association with transcriptomic behaviour. So last analysis here we did, we said, all right, so just show us the subset of genes that are both differentially methylated and differentially expressed. Let's have a look at the overlap. So for pituitary, non-significant. For liver, non-significant. Very small numbers of genes were both differentially methylated and expressed. But for the ovary, there was a significant number. So there were 195 genes that were both DE and DM. And so this prompted us to look deeper at the directionality, because for this set of 195, of course, they're both either up or down regulated, and they're both either hyper or hypomethylated. So what was the relationship of those two directionalities? And that's what this table shows. And the take home is that we see strong evidence for methylation turning genes on. So 148 of them were both hypermethylated, increasing methylation, and upregulated. And that fits this classical expectation of the position of methylation and its consequence on gene expression. So we didn't see any evidence or correlation for methylation turning genes off, but we did for increases in methylation at gene bodies, specifically turning genes on. So we did a gene ontology analysis of these 140. And what we found was very interesting. So we found a strongly enriched class of genes called the SWI-SNF and NBAF genes. And what these are chromatin remodeling complexes. So they act on the histone itself to influence more widely gene expression. So we're really interested in these because while methylation itself doesn't appear to be regulating large numbers of genes, I think it's possible that these are control points for key expression changes which are mediated through these chromatin remodeling complexes. All right, so the last part of the talk then, briefly, I'll tell you about what we found with our ATAC-seq data and chromatin accessibility changes and how that related to gene expression. So the first take-home message from our ATAC-seq data is if there are differential accessible regions, it happened early in the time course. So these are the significantly opening genome regions, or those that are losing accessibility, that were accessible and become unaccessible at T2 compared to T1. And you see there's a lot more of them, and it sort of steps down in a time-wise fashion to time point 4. So most changes occurred early, and that kind of makes sense if you think about if chromatin accessibility, and we know it does influence gene expression, it should happen before that gene expression change is realised. And that's what we saw in this data. And the other take-home there was that when things either open or regions were either opening or closing, they stayed that way with that change state for the rest of the time course. But the key thing that we were interested in was, well, what is the relationship between changing accessibility status and gene expression change? So here, the grey dots in the background are all of the gene expression data. Again, differential expression on the y-axis and average on the x. And then we overlaid on top of that differential accessibility. And this is at T2 compared to T1. And we saw really strong correlation you can see here. So chromatin state changes at T2 explain 56% of the variation in gene expression. It was about the same at T3 and T4, but a bit lower. But we see this strong bidirectional relationship between changing chromatin status and gene expression. And it was strongest for differentially accessible regions that occurred not in genes, but immediately proximal and upstream for genes. So this is the cis regulatory network. So the take home is, it appears in our data set, at least for this complex trait, that chromatin accessibility is a key regulator of gene expression, unlike methylation. The time's just about up, so I'm going to leave you with a couple of conclusions from our work. Firstly, we're really pleased that the animal experiment worked, and it was non-trivial. There was no guarantees that it would, but we're convinced that we did synchronise animals, and we did capture them at this first early stage where they entered maturation, and that's the first. Working with this multi-dimensional data taught us a lot. It was challenging in some ways, but I'd advocate it as a way forward for complex trait dissection in the future. And in particular, the attack-seq data. I just gave you a hint of it in the last couple of slides there. But if you're sitting there wondering what sort of data to generate, our experience was attack-seq was great. And the last one is where we're heading, and that's we're building towards functional assessment of some of the key targets. Some that I showed you, some that I didn't. First using gene editing in culture, and then we're going to move, hopefully, to whole animals to functionally validate some of our targets. With that, I'll stop. I'll say thanks again to these two gentlemen in particular who were absolutely remarkable to make all of this happen, Amy and my colleague Harry. Thanks, Harry. Thanks. Bye.